Hello everyone and welcome to the new session of the lecture series, and today we'll be discussing and solving the NEET previous year questions from the chapter Photosynthesis in Higher Plants. Alright, so let's start with the first question. So here they are saying that immersion enhancement effect and the red drop have been instrumental in the discovery of, so let's study what is immersion enhancement effect and red drop. Okay, so first is immersion enhancement effect. Okay, so this effect tells us that the yield of photosynthesis, you can say, okay, or you can say the quantum yield of photosynthesis, or again you can say rate of photosynthesis. Okay, the quantum yield of photosynthesis. Okay, or you can say the rate of photosynthesis, it increases. Okay, increases when the chloroplast contain the photosynthetic pigment. That's why we are talking about the chloroplast. When the chloroplast is subjected to, okay, or illuminated by, you can say, or illuminated by light, okay, of both far red and red as well, okay, or you can say both red and far red light. Okay, so there is an increase in the rate of photosynthesis when the chloroplasts are illuminated by far red and red light, both. Okay, so this is known as enhancement effect, immersion enhancement effect. And what is red drop? Red drop is the decrease in the rate of photosynthesis. Okay, beyond 680 nanometer. Okay, when we provide the light, okay, beyond 680 nanometer of light. Okay, or you can say when we provide only the far red light. Okay, so there is a decrease in the rate of photosynthesis. Okay, at far higher, longer wavelength of light, which photosystem is activated? Photosystem 1 is activated. Okay, so photosystem 1 is, okay, doesn't function very well, and there is a decrease in the rate of photosynthesis. All right, now let's look at the options. First is photophosphorylation and non-cyclic electron transport, which is not correct. Second option is two photosystems operating simultaneously. Okay, so this is correct. Okay, if we talk about photosystem 2 and photosystem 1, photosystem 2 is activated by light, or it contains reaction center with maximum absorption as 680. Photosystem 1 contains reaction center which has maximum absorption at P700. So if red and far red light are given, then there will be activation of both the photosystem, and it will lead to the increase in the rate of photosynthesis. All right, so this option is correct. Now third option is photophosphorylation and cyclic electron transport. This is also not correct. And the fourth option is oxidative phosphorylation. So this happens in mitochondria. Okay, oxidative phosphorylation takes place in mitochondria. So this is also not the correct answer. So what is the answer? It tells you about the discovery of two photosystem operating simultaneously. Okay. The next question is, water-soluble pigments found in the plant cell vacuoles are, okay, options are xanthophyll, chlorophyll, carotenoids and anthocyanin. So xanthophylls, okay, xanthophylls are not water soluble. Okay, so this is not the correct answer. Xanthophylls, okay, they are, okay, one of the compound in which they are soluble is hexane. Okay, chlorophyll are not soluble in water, but they are soluble in organic solvents. Okay, again, carotenoids, they can be dissolved in fats, and then we are left with anthocyanin. So anthocyanin are what? They are water soluble. Okay, so what should be the correct answer here? Anthocyanin. The next question is, in the chloroplast, the highest number of protons are found in, options are stroma, lumen of the thylakoid, intermembrane space and antenna complex. Okay, so we know that the highest number of protons are present in the lumen of the thylakoid. Alright, so if we talk about the chemiosmotic hypothesis, so what happens? Here we have photosystem 2 in the thylakoid membrane, then we have cytochrome B6F, here we have photosystem 1, this is the stroma, and this is the lumen of the thylakoid. This is what? This is the thylakoid membrane. Okay. So these complexes are present in the thylakoid membrane. So what happens? As the electrons flow, then what happens? Protons, okay, they are transported from the stroma into the lumen. Okay, and there is accumulation of the protons in the lumen. Okay, a proton gradient is created across the membrane. Okay, that's why there is highest number of protons in the lumen of the thylakoids. And here what becomes? There is acidic condition due to the accumulation of the protons. Okay, so option B is the correct answer here. The next question is, with reference to factors affecting the rate of photosynthesis, which of the following statement is not correct? So options are, increasing atmospheric carbon dioxide concentration up to 0.05 can enhance carbon dioxide fixation rate. Okay, option B is C3 plant respond to higher temperature. Okay, they are saying higher temperature, but C3 plant, okay, let's read the full sentence. C3 plant responds to higher temperature with enhanced photosynthesis, while C4 plant have much lower temperature optimum. So this statement is not correct. Alright, so what happens at higher temperature? C3 plants don't show enhanced photosynthesis. So this is not correct. Okay, and C4 plants have higher temperature optimum. Okay, and not lower. So this lower is incorrect here. Okay, so this entire statement is not correct. Option C is light saturation for carbon dioxide fixation occurs at 10%. This is correct. And the fourth option, tomato is a greenhouse crop which can be grown in carbon dioxide enriched atmosphere for higher yield. Okay, so option B is the correct answer for this question. Okay, at higher temperature, okay, there is no enhanced photosynthesis, but there is a decrease. Okay, now coming to the next question, which is oxygen is not produced during photosynthesis by, options are green sulfur bacteria, then we have nostoc, then we have Cara and Cycus. Okay, Let's talk about green sulfur bacteria. So green sulfur bacteria is what? It is a photosynthetic bacteria. Okay. It is a photosynthetic bacteria which utilizes what? It is sulfur. Okay. It utilizes hydrogen sulfide, H2S. Okay. For what? For conversion of inorganic carbon. Okay. Or you can say conversion of carbon into what? Carbohydrates. Alright. So here no oxygen is produced. It is what? Sulfur is produced here. Sulfur is produced instead of the oxygen. Okay. Oxygen is produced when we used water. Okay. If we replace H2S with H2O, then oxygen will be released. Okay. So green sulfur bacteria is the correct answer here. The next question is, which of the following is not a product of light reaction of photosynthesis? Okay, so during light reaction, what is formed? ATP is produced and NADPH is also produced. Okay, so options are ATP, NADH, oxygen and NADPH. Alright, oxygen is also produced. Where? Oxygen is produced due to the photolysis of water. Okay. Which takes place, what is the site? Photosystem 2. Because photosystem consists of the water harvesting complex. Okay. So, we are left with the NADH. Okay. So, if we are talking about NADH, we are talking about mitochondria. Alright. So what should be the answer here? Not the product of light reaction. Option B, NADH, is the right answer for this question. Now, let's come moving on to the next question, which is the oxygenation activity of Rubisco enzyme is in photorespiration leads to the formation of. Okay, so they are asking about the first product which are formed during the photorespiration. Alright. So now you know that Rubisco has both. If we talk about Rubisco, it has both carboxylase and oxygenase activity. Carboxylase as well as oxygenase activity. Okay, means it can bind to carbon dioxide also. It can bind to oxygen also. Okay, or it can catalyze, you can say Rubisco can catalyze the conversion of, or you can say the assembly of carbon dioxide with RUBP. Okay, and the assembly of oxygen with RUBP as well. Ribulose 1,5 bisphosphate. Okay, now they're talking about photorespiration. So if we talk about the oxygenase activity of Rubisco, okay, then we are talking about the photorespiration. Okay, so what are the products here? When Rubisco binds with oxygen instead of the carbon dioxide, the products are one molecule of 3-phosphoglyceric acid. Okay, a 3-carbon compound is formed, 3-phosphoglyceric acid, along with the 2-carbon compound phosphoglycolate. Okay, phosphoglycolate is also produced, which is a 2-carbon compound. So here in the option they are saying one molecule of 3-carbon compound. Okay, then one molecule of 6-carbon compound, not correct. Then a molecule of 4-carbon compound, again not correct, and 2 molecules of 3-carbon compound. This is also not correct. Okay, one molecule of 3-carbon compound should be the answer here. Okay. The next question is, in light reaction, plastoquinone facilitate the transfer of electrons from Okay, these are the options: cytochrome B6F complex 2, photosystem 1, photosystem 1 to NADP plus photosystem 1 to ATP synthase, and photosystem 2 to cytochrome B6F. Okay, so let's talk about the photophosphorylation or the electron transport chain. So what happens here? We have photosystem 2 here, and then we have photosystem 1 here. So this is what? This is non-cyclic photophosphorylation. Okay, phosphorylation. So what happens here? Photosystem 2 absorbs the photon, it gets excited. Okay, P680 in excited form. Primary receptor is the pheophytin. Okay, pheophytin transfers electron to plastoquinone. Plastoquinone transfers electron to cytochrome B6F. Okay, and then we have the plastocyanin here. Plastocyanin finally transfers electrons to photosystem 1. Okay, and photosystem 1 also gets excited. Okay, here we have P700 in excited form. Here we have ferredoxin reducing substance as the primary acceptor. Then we have ferredoxin. Okay, and finally what happens? Electrons are transferred to NADP plus, which is converted into NADPH. Alright, so what should be the answer here? Plastoquinone facilitates the transfer of electrons from, so this is plastoquinone, from photosystem 2 to cytochrome B6F. So option D is the correct answer here. The next question is, which of the following statement is incorrect? Alright, so first statement is cyclic photophosphorylation involves both photosystem 1 and photosystem 2. Okay, in the previous question only we have seen that non-cyclic photophosphorylation, okay, involves both photosystem 1 and photosystem 2. This is incorrect statement. Now next statement is both ATP and NADPH are synthesized during the non-cyclic photophosphorylation. This is correct. Okay, both are produced. Okay, now third option is stroma lamellae have photosystem 1 only and lack NADP reductase. Alright, this statement is correct. Okay, this statement is also correct. Option D, grana lamella have both photosystem 1 and photosystem 2. Okay, so this is also correct. Here non-cyclic photophosphorylation can occur. Okay, non-cyclic photophosphorylation will occur. Here what will occur? Only photosystem 1 is involved, so cyclic photophosphorylation can occur here. Okay, and why? Because it lacks the NADP reductase enzyme also. Okay, that's why only cyclic photophosphorylation will be activated. So what should be the answer here? Option A. This is the incorrect statement here. Alright. Now next question is the first stable product of carbon dioxide fixation in sorghum. So sorghum is what? Sorghum is a C4 plant. Okay. The first stable product they are asking. The first stable product. So in C4 plant what happens? Carbon dioxide is accepted by the prime receptor phosphoenol pyruvate, which is a 3-carbon compound. Alright. Then what is formed? Oxaloacetic acid, which is a 4-carbon compound, is the first stable product of carbon dioxide fixation in the C4 plants such as sorghum. Okay, other examples are maize. Okay, one more example of C4 plant is maize. Okay, so here the options are phosphoglyceric acid, which is not correct. Pyruvic acid, not correct. Then we have oxaloacetic acid, which is correct. And the fourth option is succinic acid, which is not correct. Alright. So what should be the answer? Oxaloacetic acid, which is a 4-carbon compound. That's why the C4 plants have been given the name C4 also, because a 4-carbon compound is formed. Alright, and this reaction is catalyzed by which enzyme? PEP carboxylase. Okay. Alright, now let's move on to the next question, which is which one of the following is not true regarding the release of energy during ATP synthesis through chemiosmosis? Okay. So chemiosmosis, okay, we already know that chemiosmosis is related to the generation of ATP by the breakdown of the proton gradient. Okay, first there is a development of a proton gradient as the electron move from one carrier to another carrier, okay, or as the electron move downhill. All right, and this, okay, breakdown of the proton gradient later leads to the ATP synthesis. All right. So first option is breakdown of the electron gradient. So this is not correct. Okay, so we have got the answer here. Option A is the answer here. There is no breakdown of electron gradient. There is only breakdown of the proton gradient, which leads to ATP synthesis. Second is movement of the proton across the membrane to the stroma. So this is also correct. Option C is reduction of NADP to NADPH on the stroma side of the membrane. This is also correct. Okay, this takes place. What is the site for this? Photosystem 1. Okay, which contains the enzyme NADP reductase. Now, fourth option is breakdown of the proton gradient. So this is also correct. Okay, so the statement which is not true, okay, which should be option A, breakdown of the electron gradient. The next question is, what is the role of large bundle sheath cells found around the vascular bundles in the C4 plants? Okay, so these bundle sheath cells, okay, they contain large chloroplast, large and agranal chloroplast. Okay, so let's look at the options. First is to increase the number of chloroplasts for the operation of the Kelvin cycle. Alright, option B is to enable the plant to tolerate high temperature. This is not true. Option C is to protect the vascular tissue from high light intensity. This is also not true. Fourth option is to provide the site for photorespiratory pathway. This is not true because the C4 plants, okay, what they do, they avoid photorespiration. Or you can say they have reduced photorespiration or they lack photorespiration. Alright, so what should be the answer here? To increase the number of chloroplasts for the operation of the Kelvin cycle. Okay, these bundle sheath cells, okay, contain large chloroplasts and they increase, okay, and they help in the operation of the Kelvin cycle. The next question is, two statements are given. Okay, statement 1 and statement 2. Statement 1 is the primary carbon dioxide receptor in C4 plant is phosphoenol pyruvate, PEP. Okay, and is found in the mesophyll cells. So this is also true. Okay, this statement is correct. Then statement 2 is mesophyll cells of C4 plant lack Rubisco enzyme. Okay, this is also true because mesophyll cells contain which enzyme? PEP carboxylase. And Rubisco is present where? Rubisco is present in the bundle sheath cells. Okay, where they are involved in the C3 cycle. Okay, so now in the light of the above statement, choose the correct answer from the options given below. Both, okay, first option is both statement 1 and 2 are incorrect. Not, okay, this is not correct. Option B, statement 1 is correct, but statement 2 is incorrect. This is also not the answer. Option C, statement 1 is incorrect. Okay, again, this is not the answer. And then option D is both statement 1 and statement 2 are correct. So this should be the answer here, option D. The next question is, the reaction center in photosystem 2 has an absorption maxima at, alright, they are talking about the photosystem 2, okay, what is the reaction center of photosystem 2 made up of? The reaction center is made up of a single chlorophyll A molecule which has maximum absorption at 680 nanometer. That's why denoted as P680. Okay. So the options are 680 nanometer, 700 nanometer, 660 nanometer and 780 nanometer. So what should be the answer? 680 nanometer. Alright. The next question is, which of the following combination is required for chemiosmosis? Okay. Options are membrane, proton pump, proton gradient, ATP and ATP synthase. Next option is membrane, proton pump, proton gradient and NEDP synthase. NEDP synthase is not required, so this option is not correct. Option C, proton pump, electron gradient and ATP synthase. Electron gradient. We require electrochemical gradient. Okay, this option is also not correct. Then there is no membrane here. Membrane is also required. Okay, so this option is not correct. And the fourth option is proton pump, electron gradient and NEDP synthase. Again, this is not correct. So we are left with option A. Option is membrane. Yes, membrane is required because proton gradient is formed across the membrane. Okay, and membrane is also required because complexes are present in the membrane, thylakoid membrane. All the complexes are present, photosystem 2, photosystem 1, okay, as well as the ATP synthase is also present in the membrane, and then we have cytochrome B6F is also present in the thylakoid membrane. Then proton pump is required, yes, so that the proton gradient can be established. Proton gradient obviously is required, okay, because breakdown of proton gradient later will lead to the ATP synthesis. And ATP synthase, what is? It is involved in ATP synthesis. Okay, as the proton moves back into the stroma through ATP synthase, then the generation of ATP from ADP takes place. So what should be the answer? Option A is the correct answer here. Alright, so this was the end of this lecture. Thank you very much for listening.